Hold on. Go. Oh, hey. Didn't see you there. My name's David Becker, and I'm your CA /3LPC, and welcome to Exam 3 Review. Today, we're going to be able to over all your questions, so come to me, curious, and get ready. But make sure, before you ask the question, make sure you find it. Look at this picture. So dumb. You're welcome, Alejandro. All for you. You get that privilege.
You got 13 people watching.
13 people.
Okay, so
Let's make it happen.
just that it's student-led and to ask you questions.
Okay, so the difference between this CSS, apart from the other online CSSs, is that this one is going to be guided by you guys. I don't have an agenda. I don't have anything scripted to say. You guys come with questions. I come with answers. So shoot me questions.
A couple of them have left to sign in. I think it's usually what happens.
Mm-hmm.
You post the link. We got 8. That's the highest number we have.
Hey, get it popping. Back to back. Watch one of them be like Carrier David.
Yeah. Oh, there's a question. What are the different points of regulation in eukaryotes for what? You see it?
Mm-mm.
See the question?
What are the different points of regulation in eukaryotes? In terms of regulation?
I believe it's cell cycle.
For cell cycle?
I think so.
Well,
And then Christopher asks, can you go over cyclins and CDKs? So I'm assuming those are both connected.
okay.
So we can just talk about that.
So seems like we want to go over cell cycle. So let's go over cell cycle. So knowing the cell cycle, let's go over let's split up the cell cycle into the different phases. So in the cell, we have G1. S, G2, and what?
Yeah.
That's right, M. But M is split into 2 phases. What are the 2 phases? Answer the question in your comment. Ooh, advantage. What's M represent? Type it out for me. It's interactive.
Yeah, they don't. This is happening every time.
Where?
Yeah, we got it.
We got it?
Nice. We got some.
What?
You see it?
Mitosis. Mitosis. It's mitosis. Mitosis or meiosis.
Okay, Alejandro, we see your question. We'll get to it after we do cell cycle stuff.
And so, within mitosis and meiosis, there are 3 checkpoints. One checkpoint is within G phase. Checkpoint. And then the other checkpoints are at the beginning. One's at the beginning of meiosis and mitosis, and one is at the end of meiosis and mitosis. And so, it's backwards when I type? Like, is it backwards for them as well?
No. What do you mean?
As in, like,
it's like a mirror image, yeah.
Mm-hmm. Is it a mirror image for them as well?
No. I can see fine.
Okay.
Yeah, they flipped it.
Nature. Okay. So meiosis and mitosis. Those are the 3 checkpoints. And so, in terms of CDKs and whatnot, so as we progress through the cell cycle, specifically like starting from G1, as we go through, after the synthesis of DNA, after the synthesis, which is the replication, cyclins start getting created. And as we are approaching G2, if there's enough cyclins once there are high concentration of cyclins and MP what again?
How much more cyclins bind to CDKs?
CDKs, right? Okay. Once enough cyclins bind to the CDKs, we create our MPFs. Does that make sense? So, as we go from here to here, cyclin concentration increases. When the cyclin concentration increases adequately and they bind to enough CDKs, which are cyclin-dependent kinases. Those bind together to create MPF. MPFs specifically once hitting metaphase of either meiosis or mitosis dictates if it keeps going. And after the cell replicates and cytokinesis occurs, the CDK, the cyclin on the MPF start to break down and degrade. And what we have left is CDKs, and those CDKs are going to be recycled again to go through the process of the checkpoints.
So we've got a new question. What's up, guys? Okay, so just a quick little example. You see the MPK cyclin CDK complex. So your cyclin's going to bind to your CDK, and that's basically like going to form like a megazord kind of thing that'll push the whole process into the next thing. So that's how you can control it. But if you can control this, the cyclin, from binding to the CDK stands for cyclin-dependent kinase. So it'll only work when you have this guy bound to this guy, right? So once you have both of them, that's what can push it forward. And then Jessica has a good question too.
And Jessica, how does the regulation of gene expression differ between pro and eukaryotes? It's a good question.
Is that what like operon? Okay, that's okay. Start. Use a different color. Use a glare. Let's go.
Oh, we're good? Okay. So the difference between pro and eukaryotes. So if you remember in class, for prokaryotes, main point of regulation for gene expression is the operon. Now, can you guys tell me in order in your chat box, in the chat box? Can you guys give me the order of the operon and what it consists of? The order of components of the operon.
Okay, so Jessica has a good question. Can you hear us at basal expression?
Yeah. Mm-hmm. Yeah, of course eukaryotes have basal expression. If you remember what she was talking about in class, and this applies for both pro and eukaryotes. In terms of expression, there's never a lull or there's never a complete shut off of the gene. There's always, instead of a shut off, it's just a low expression. So just like we're talking
absolute zero.
Yeah, exactly. It never if you guys remember from chemistry, for in terms of the physical state of things, when you bring something to absolute zero, molecules stop moving. that's impossible. That doesn't happen here. What happens is you just bring it down low enough to where it's the gene of expression is negligible. Promoter,
We've got some answers.
operon, and genes. Okay, Joanne. Okay, Joanne. Okay.
Okay, you have to give him a while after you ask me a question because there's a lot of lag. I'll make it. I'm finding another part.
Cool. Cool. Man, this David guy. Whoa. Man, this David Hernandez? David Hernandez. I don't think I've seen him on the roster.
Oh no.
Okay.
I think he dropped.
So, yes, the prokaryotes, on the DNA of a prokaryote, it contains a promoter, an operator, and then the genes. So, can you guys tell me about the two types of regulation in terms of operons? I'll give you guys some time. Let me just roll off.
Can you mute it?
Just push the mic.
Yeah. Cool.
I'm back. And that was, you guys, man, Alejandra and Joanna are on this. Got negative. So we got,
Let me see if I can turn up.
apologize for some technical difficulties. We got negative and positive. All right, good. And now in terms of negative and positive regulation, if you guys, well, now for a second, we'll come right back to prokaryotes. Let's go to eukaryotic expression. So for eukaryotes, can you guys also give me the order of the components of the DNA for eukaryote? I'll be right back. I'll let you guys do your thing. I'm back, and let me see. So we have enhancers, promoters, control genes, maybe by Joanna. And Crazy for Pizza says activators, enhancers, promoters, and genes. And I like those. I like those. So you guys are both correct. But specifically for Crazy for Pizza, I'm gonna need to ask you a follow-up though, just to clarify. So the question was, what are the components of on the DNA in terms of for gene regulation? And we'll come back to your chat comment in a bit. But yes, we are gonna break it down. So we have our enhancer sequencing. Let me just color them so it's easy to. And this guy, just to see, is the glare messing with you guys? Is the glare annoying in terms of the board? Comment, let me know.
Anytime you need a picture or anything, let me know.
Enhancer. Promoter. And genes control.
I think you're good. You just need to write a little bigger.
Yeah.
Next time.
So going back to Crazy for Pizza. Crazy for Pizza said activators, enhancer, promoter, and genes. So three-fourths of that answer is right. We do have an enhancer, we do have a promoter, and we do have the control gene. But the activator is not a component of the DNA. If you guys remember me talking about this in class, this will mix you up. It's because of activator and enhancer. That's what's going to mix you guys up. So make sure you can distinguish.
Yeah, you can write a little bigger. That's good.
These are the things that are going to mix you up. So E means, I can't think of a good mnemonic right now, but E means it's going to be placed on the DNA. OR is not on the DNA. Just remember that. But yeah, the activators are going to be the transcription factors within that respective cell. If I know what I call it. That bind to the DNA. And once bound, initiates the next phase of regulation. And now somehow these activators need to go and touch these control factors in order to start the process of regulation. So what are the molecules or proteins necessary for this to happen? I'll let you guys.
Wait, hold on. Time out. I found a really terrible mnemonic. So the OR, if you extend that R, you can make it an N. So activators can bind on the protein or on the DNA. The proteins bind on the DNA. Enhancer has an E in it, and so does sequence. So enhancer sequences of DNA.
I'm back. How are you guys? Good to see you guys. So let's check through this chat. Thank you, Asinia. But you're right. I'm not grading the papers. And so so Kevin Moy. Yeah, DNA bending proteins and mediator proteins. That is correct. That is how we're going to get these activators onto the control genes. And now, just to dispel some confusion, you thought control genes were binding site. Alejandra says she thought that control genes were the binding sites for the activators. So, in this case, the enhancer sequence, I think you're getting the control genes and the enhancer sequences mixed up. So, in terms of the control genes, the control genes are the genes we're literally trying to control and regulate. The activators bind to the enhancer sequence, and the sequence technically it's a sequence of DNA, which is it's like a gene, but in this case, the purpose of the sequence of the DNA is for the binding of the activators. Does that make sense? Let me take that as
Oh, also we got that one. Are there activators?
And then Joanne is asking, is there activators in prokaryotes and eukaryotes? Okay, this is a good question. This is a very good question. It's a distinguishing question. Now, before I answer your question, I want to make sure you have this correct. Let's think about eukaryotes. Eukaryotes, most of the time, are big multicellular organisms who contain different types of cells. Although all those cells have the same DNA sequence, somehow each cell has a different function. So, what molecules are different within the different cells that help to differentiate expression amongst all the cells? I'll wait.
The whole thing takes a while. Like 10 seconds. Okay. I'm just gonna write down some of the topics I want you to hit. Regulation of oncogenes. Just a quick aside, we are taking note of some of the questions that we haven't talked about in the chat because a lot of the questions are just grouped together, so it just makes sense to go over a certain few in a particular order, but we will get to these. Thank you for asking.
So the nth that I was looking for is transcription factors. Within eukaryotes, especially multicellular eukaryotes, in order for them to differentiate expression in their different cells, they have different transcription factors. Prokaryotes, on the other hand, are often unicellular. Since they're unicellular, they don't have to actually differentiate expression. So to answer your question, whose question was this again? Joanna?
Yeah.
There are only activators in eukaryotes. But going back to this, as my boy Kevin said, we have, I'm not going to draw out the whole thing. I'm not the best drawer. But yes, we have our DNA bending protein. In tandem with our mediated proteins, which create a protein complex that aid in this. And what happens is that they fold over, but with the aid of the DNA bending proteins and the mediator proteins, it's going to bend the DNA and put these activators onto the genes. Once the activators connect to the genes, then what's going to happen is that there's going to be I apologize. The activators don't connect to the gene. The activators are going to go and connect to the mediator proteins, and the mediator proteins are going to connect to where the promoter is. And what's going to happen is the activators plus the mediator proteins on top of binding at the promoter, it's going to create a complex. That complex is where the RNA polymerase is going to bind. RNA polymerase 2 is going to bind, and it's going to create the RNA that's necessary for the expression of this gene. Jessica, how can multiple? Jessica's asking, how can? Well, before Jessica's question, let's make sure we.
Yeah. Let's see. Really quickly, Jessica, I can answer that one. So you remember in prokaryotes, you don't have a nucleus, and so you like think about the timeline between transcription and translation. Everything happens pretty fast. So if you can see from David's drawing here that you forgot your mRNA, but you've got 3 genes coded on there. So basically, sorry, this is your DNA. And then you're gonna get 1 transcript, right? And so basically you're gonna have a ton of, you have tons of ribosomes on that mRNA at the same time, and those are going to begin translating. That's all the stuff that we know, right? You need to have your thing. I would assume that within this mRNA sequence, you have a start, so it's AUG, and stop. So you basically just have 1 long transcript with multiple like sites for ribosomes, and then within that, you're going to have multiple start and stop codons, which can give you different proteins. Does that make sense? But the key thing to remember with that is because there's no nucleus, everything is right next to each other. It happens very fast. It's what we call coupled, if you remember from exam 2. And then, okay, so yes. We had a question a while ago from Alejandro, about the events of synaptic signaling, and Dave's gonna come back.
It's your boy David back at it again. So the events of synaptic signaling. So that was Jessica's, correct? Alejandro. Oh, Alejandro, can you actually specify what do you mean as in the question? So do you mean like what's literally happening at the synapse? Or do you mean like what's happening within the neuron itself, along like the membrane of the neuron?
Maybe she'll have.
What happens after it goes across the synaptic cleft?
Okay. She's talking about like the length. Start from calcium, the voltage gain calcium, and then the synaptic vesicle leaks. How you got this?
So,
can you draw a bigger vesicle?
Bigger?
Yeah, bigger vesicle. You gotta really exaggerate.
So, if you guys remember, when we have our action potential, the action potential, what's going to happen is going to travel down the membrane, and that change in potential is what's going to happen is going to hit the voltage-gated calcium channels in the presynaptic cell. When it hits the voltage-gated calcium channel in the presynaptic cell, what it's going to do, it's going to create an influx of calcium inside the cell. That influx of calcium, that increase in the calcium concentration inside the cell, is going to attract vesicles, which are readily holding neurotransmitters. They're going to attract the vesicles to the membrane. And once the vesicle fuses with the membrane, it will release neurotransmitters. with the release of those neurotransmitters, what may happen, one of the many things that can happen is that it will bind to a receptor. Make that a bit bigger, because yes, it's going to bind to a receptor, and that receptor will then have some specific function in the postsynaptic cell. So in this case, most of the time, when a neurotransmitter binds to the postsynaptic cell, it'll actually create a new action potential in that membrane, by causing an influx of what ions? Salty banana. You know exactly. Salt ions or sodium ions. Influx of sodium ions. So with the binding of the neurotransmitter to the postsynaptic receptor of the postsynaptic cell, will activate, will actually open up sodium ions in the postsynaptic cell, which causes an influx of sodium, which then raises the membrane potential and causes another action potential to occur. So, okay, but Joanna, Alejandro, does that make sense?
Okay. Okay, yeah. So just to add on to that, similar to sodium, calcium, there's a high calcium concentration outside the neuron. So once the action potential signal hits this voltage gated channel, well, it's voltage gated, so you know, like once it hits a certain voltage, it's gonna open. It opens, and we know that we have a high concentration of calcium outside. That once it opens, calcium is gonna flood in, and then once calcium comes in, my physiology professor called calcium like a 2-year-old with like a little marker running around in your house. It's gonna like make a mess and do everything. So calcium is gonna signal like a mess of signaling events, which will eventually cause these vesicles to be release neurotransmitters into your clefts right here, which will then bind to your receptor, and these receptors will open ligand-gated sodium ion channels. I believe you have to talk about what kind of channels you know. Ligand-gated is one of them. So there's that. And then, do we have another question? We do not. Okay. Typing back out. All right. Okay. So done with that. We have gene regulation for oncogenes and glycoprotein.
The difference between a glycoprotein and a glycolipid. So, as you can see from the prefix, this is going to be Joanna Gardner's question, a glycoprotein and a glycolipid. So yeah, a glycoprotein, as you can tell from the prefix of it, means that it has a carbohydrate connected to it. So glycoprotein is a carbohydrate and a protein, which is usually a polypeptide sequence, which is made up of amino acids. Glycolipid is usually carbohydrate connected to a lipid. Mm-hmm.
What is that question?
What's the difference of their function in the I don't know, glyco. Well, we're gonna look it up for glycolipids, because right now we both can't remember. Definitely for glycoproteins, their function in the membrane is used for cell to cell identification.
Yeah, it's like your blood. I think you might have to ask the chat what question about.
Right now, just to update, could you guys just re-ask any questions that you guys had before, that wasn't answered, just to make sure.
How gene expression works. It's married.
So, in terms of gene, so I'm thinking with gene expression, like the coiling of like chromosomes and stuff?
I guess prokaryotes here, it's two different answers.
Yeah.
I mean, the overall thing is like you want to control it, right?
Mm-hmm.
Yeah. So typically, you start controlling mRNA transcription.
So for gene expression, as you might have heard Derek whispered, there is a big difference between it in terms of prokaryotes and eukaryotes. But mainly for gene expression, one of the primary levels, like the most effective levels to regulate a gene, is actually through mRNA. So whenever you create the mRNA, you can either promote with that mRNA. Yeah, with the mRNA, with creating that mRNA, you can either promote the creation of it by, in terms of eukaryotes, splicing them and condensing them into their exons, and then from there creating their proteins, or you could actually create the mRNA and degrade it.
All right, we're going to take a quick break. We're going to come back with proto-oncogenes and tumor suppressors. All right, so we had a question a while back about proto-oncogenes versus tumor suppressors, and okay, yeah. So let's start off with tumor suppressors because I think that one's just straight up easier. Like, think about the name, tumor suppressors. So that's going to be a protein that goes back and fixes corrections within the DNA, or it senses basically like tumor, like, does it sense like if cells are growing really rapidly, they'll just kill them?
For tumor suppressors?
Yeah.
Yeah, those are usually the regulators.
Yeah, so basically it's like regulation proteins, right? So if something's growing out of control or something's mutating the DNA, it will go and kill that thing. So if it's already messed up, you basically go in and kill it so it doesn't keep spreading, right? So that's a tumor suppressor. The name is really familiar. So for proto-oncogenes, if you look at the prefix proto means before, and onco means cancer and then there's gene. So it's before it becomes a cancer gene. So basically what that is, proto-oncogenes, remember that P in proto, you can think of it as terrible potential. So they have the potential to become cancer genes, right? And so basically when those genes are mutated, they can cause the cell to grow rapidly and continuously divide and basically like grow out of control like cancer that we know. So for those genes, they're typically the ones that regulate the cell cycle. So if you want to think about like genes that control the cyclin, the CDKs like we talked about earlier, remember that the cyclin, the cyclin binds to the CDK and pushes the cell through the cell cycle. But what happens if their cyclin's mutated and it can't come off of the CDK? So it's going to constantly push the cell through cell cycle when it's not there. And when you're constantly doing that, you're making the cell divide, and that's basically cancer. So the way I like to remember that is there are two ways to get in a car accident. You drive really fast or you don't brake. So you drive the normal speed and don't brake. So the proto-oncogene is when you go too fast. So, right? So you're going too fast and you keep telling yourself to divide because you have a mutated gene that tells you to go forward. And your tumor suppressor gene, well, that's your brake, right? So if you cut off your brakes, you're going to get an accident. And the two ways to get an accident, like an accident means cancer. So once again, proto-oncogene, you're going too fast on the gas and you hit the wall as an accident because you're dividing too much. You press up your brakes, aka your tumor suppressors, you can't stop what's already mutated, so you're gonna crash again. Hopefully that makes sense. If you guys need me to clarify, please let me know in the chat. Thanks, Lila.
So, independent assortment. We have this question: what is independent assortment? So, in the cell, usually for eukaryotes, especially for diploids, we're gonna have
Water.
yes, please. Mm-mm. Our chromosomes. As you can see with our chromosomes, this is going to be a diploid cell, so we know that this set of chromosomes is from the mom and this set of chromosomes from the dad. And then in this case, we have two sets of chromosomes. So this is one set of chromosomes. It's two sets of chromosomes, two different chromosomes, the long one, the short ones. So let's say if I say that these are labeled as D, D, L, big L, and little L. What the law of independent assortment says is that the separation of these specific chromosomes will not affect these chromosomes. Now, what I mean is that in this case, let's say we're going to NFA 1. We have a separation of our homologous pairs. I'm going to put little d over here. And then we got big L, and we got little L. Big D over here. And then what we're going to do is go to anaphase 2. Create our gametes. Apologize for the drawing. It was a little bit tough to see. And if we were to drop the the genotype of each one, we have L, we have little L, big D. Oops. Little L, big D. Big L, little D. And big L, little D. So ultimately, what it's saying is that the separating of these homologous pairs don't affect the separating of these homologous pairs.
Yeah, and just to add on to that, okay, so you guys see how it's kind of hard to see, and we apologize for that. Our markers are garbage, but you see how it's orange and blue orange, the different colors representing the different parents. And you see the longer sticks versus the shorter sticks. These represent different chromosomes. For example, chromosome 1 and chromosome 2, right? And basically what David drew is, do you see how on this side we have blue, orange, blue, orange. So blue's on one side, orange is on the other side. So that's one possible combination, right? We could also have, let's see, blue, orange, orange, blue. So basically on the left and right sides of this cell, it doesn't matter if it's blue or orange. And remember, there's multiple chromosomes. So we're showing only 2 for simplicity's sake. But it doesn't matter if it's blue or orange on the side. As long as both sides, when we split it through meiosis, excuse me, as long as each subsequent daughter cell has a chromosome 1 and a chromosome 2, because obviously that makes sense because you need all of your chromosomes for their proper instructions in your life. Yeah, so it could be blue, orange, orange, blue, orange. It doesn't matter. And if you look at statistics, it'll pan out. You'll get to this more in genetics. I don't think Dr. Latham focuses too heavily on that. But independent assortment is they assort. I mean, they go left and right from meiosis independently. It doesn't matter. They have no effect on each other. There's also a really good picture in the book. That's you can see.
In transduction, what proteins can affect? So Karen Nunez is asking, in transduction, what proteins can affect the pathway? Pathway.
Oh, it's like that activity. Okay, so Karen, to answer your question, what proteins can affect the pathway and how they're turned off? So let's think about that for a second. So it's been a while since we had a collaborative question. So in the chat, I want you guys to think or just jot down some possibilities of what you think might be able to turn off a pathway for, let's say, a G protein. And we'll give you guys just a second. Okay, so Joanna had a good response. It says G protein can turn it off. So yeah, so when the G protein has a bound GDP and gets displaced back with the GDP, it's no longer active. So we know that part's off. So my question for you guys now is that the only way? Yeah, so we see a lot of questions about changing DTP back to GDP in the G protein. That's definitely a way, but is that the only way? And Crazy for Pizza says a mutation in adenyl cyclase. I'm assuming you mean to mutation as a non-functional, and that would also be correct. But for this case, we're talking about a completely normal pathway. Actually, it's just turn the pathway off. Yeah, that's definitely one of the reasons. You can have a mutated protein within the pathway. So good job, Crazy for Pizza. But can you guys think of any other reasons? This might be a good exam question. She might say, "Oh, list a couple of ways the pathway could turn, or areas where the pathway could be turned off."
Definitely.
So we'll give you guys another second. Yeah, exactly. So if the signal leaves the receptor, then you're going to turn it off. Definitely another solid option. Nice. Crazy for Pizza says phosphodiesterase, not turning off the kinases. So be careful with that wording yet, because phosphodiesterase does not turn off kinases. What does that turn off? Can we get in the chat? It's over here for you guys. Yeah, so phosphodiesterase actually it breaks down cyclic AMP, so CAMP. And the other really annoying thing is, so the thing that takes away phosphates from active proteins is what?
Mhm.
Yeah, phosphatase. Nice job, Jessica. So, crazy for pizza and anyone else confused by that. So, phosphodiesterase will break down CAMP, and phosphatase will break down phosphates. And we mostly is just all phosphate with a P and a circle in it. Yeah, so those are two different things. I would focus more on phosphatase, just because she doesn't talk about this one too much. But you can see how that's both important, right? So if we just if we take out the phosphate and we deactivate that one particular protein, well, that's fine. We stopped the pathway momentarily, right? But if we still have some CAMP floating around, we can reactivate the pathway at the start, and that's going to trigger another signal cascade to basically turn that protein back on again. So not only do you need to turn off the individual proteins, you need to turn off the source that's activating all the kinase cascades, which is the cyclic AMP. Does that make sense? Okay, so Alejandra asks, what is the role of AMP? Honestly, I don't know. You don't have to worry about that. So the importance of cyclic AMP, so it's like the C, it's cyclic. Okay, obviously, so AMP is in a ring, and it's like forced into a ring. And when molecules are forced based like in like certain positions, they have a lot of energy. So once you break that ring, you can use that energy to power things. So that's how that first kinase gets activated when cyclic AMP is bound to it, and it'll break, and then it'll basically provide energy for it. You'll learn a lot about that in OChem and in biochem. So obviously, once cyclic AMP is broken, it's no longer cyclic, so it's just AMP, and then I'm sure other metabolic processes can happen to AMP. But yeah, it's just that whole really like cranking it into a ring is what gives it energy. Don't worry about that too much, but that is a good question. And then if you guys haven't already signed in the link in the description so we can get paid. We put a lot of work into this with the intro and stuff.
Any more questions? Throw them at us. And give us feedback. If something doesn't make sense, say it doesn't make sense.
Yeah, if you like David better than other David, let me know.
Don't let him know.
He doesn't watch these. Okay, so we've kind of stopped with the whole backup questions. We've caught up to all your questions. And also when you're studying, keep in mind, like, so Dr. Latham gives you a lot of proteins, especially in like signaling. I think this is where this is most applicable. Signaling and operons and like regulation as a whole. So think about each protein and what is a potential scenario, or like what would happen if that protein is mutated. So practice question, let's say an activator protein for
Liver.
your liver. Oh, wait. Okay, so let's say the protein that breaks down, that helps detoxify alcohol in your liver is mutated, right? And so it's a mutation, so it's not functional, and it's mutated so it can't bind to its enhancer sequence. What may be a potential cause of that? Okay, so we're gonna move on because we've got some good questions. Meredith asked about the gradients. So gradients basically just means there's like two different amounts of something on either side of a membrane, yeah, or something. So we've got two types. We've got concentration and electrical, and we'll talk about concentration first. So we have. It's pretty same. So let's do a vertical. Okay. Wow, that's terrible. I'm so sorry. So bad. I'm so sorry. Okay. So let's say you got a container that's separated by a wall, and it's got various amounts of H2O in the two compartments. Your two compartments, right? So what's going to happen when we just open the gap between the compartments?
Derek, I think I know.
Oh yeah, what is it, David?
I think that water from this side is going to flow over to that side.
Oh yes, exactly. So water and yes, Meredith, it'll be equal. Yeah, so exactly. Water will flow from the higher side, the higher concentration of water, or the higher amount, to the lower amount. And so eventually, Meredith, you are right that they will be equal. Right? See, if you want to know something about, like, this is just a big theme for sciences and upper division classes in general, nature hates gradients. So it doesn't like this. It always wants it to be equal. Right? And so, and think about, because it's water, right? Think about like hydroelectric dams. You can have like a motor in here that as it flows through, you can power that, or as the water flows through, it can spin the motor. It can use it as power to do something like make electricity or whatever. But yeah, so nature always wants to make it go to this one, even exists as this one. So where is a good example of where we see some gradients or concentration gradients? Concentration just means like different physical amounts of something. That's that type of gradient. But where's a really good example where we've seen this kind of gradients before? Hey David, do you know where we see gradients a lot?
Let me think about it. Oh, neurons.
Oh yeah, not squid, neurons.
Oh.
Okay, so if you recall, we see various amounts of concentrations of ions on either side of it. So what do we remember is high on the or outside?
You remember it. You remember it. Say it. Say it. Later. No. No.
Okay, yeah. So we see a lot of sodium on the outside, and that's just it's just how it is. So we have a high extracellular concentration of sodium, and just a little bit on the inside. So like that water example, what happens if we just open up an ion channel? What do you think is going to happen to that sodium there? Come on, guys, you're doing so well in the beginning.
I believe in you. Yes, Kevin.
Yes,
coming
Kevin.
through.
My boy Kevin. Yeah. All right. So exactly the sodium, because nature hates gradients, right? So we have a little bit on the inside, and all these sodiums are going to rush in until the concentration sodium out equals concentration sodium in. Right? And then what about potassium? Where do we see that? What's the whole deal with potassium? Come on, Kevin.
K is mainly inside.
Nice, Kevin. I love you right now. Yeah. So K is you have a high potassium potassium concentration inside the neuron, inside most cells too. What do you think, physio? I think it's
That's true.
yeah. Yeah. So just like Kevin said, yeah, and Meredith, we'll get to the charges in a second, but we have a high amount of potassium on the inside. So exactly like before, what happens when we open that channel, that potassium is going to move towards the lower concentration, remember high to low, and that's going to flow outside of the cell. So that's a concentration gradient and a good example of where we've seen it. I'll let you absorb that squid looking neuron. Okay. And then now we're going to talk about electrical gradients. And I think, Meredith, this might be where you wanted to talk about. But basically, that's the same thing, except this time we're not talking about physical amounts. Well, kind of. But we're talking about the amount of charge something has. So we're not looking at, let's say, neutrally or nonpolar objects. We're typically talking about things that have a charge in that container. Let's see. Okay, so basically we've got sodium ions in one container and chloride ions in another. So obviously if we separate the overall charge of this compartment, that'd be positive. That makes sense, right? Because we just got a bunch of positive ions in there. And then this one would be negative. So, there's a difference, there's a gradient of charge, right? Difference of charges, right? So just like, think like super logically, what would happen if I just open the containers to each other? What do you think is going to happen? Let me know. Yeah, exactly, Meredith. So the charges are going to fly towards each other because positive and negative are attracted to each other. Opposites attract. Nicely done. Okay. So that's like the basis of a gradient. You don't have to know super in depth for your exam, but hopefully I want to explain something that'll make sense later. So when you have, let's just say a membrane of a cell right here in the inside, typically it is negative. So I do a bunch of negative charges here. They're not, it's a non-dashed line, it's negative charge. And well, let me ask you this. So what would be more likely to flow, ignoring amounts of concentration, right? If I have a positive ion and a negative ion, which one of those do you think will have an easier time going through the membrane? Let's give each of them a channel. So let me know in the chat what you guys think. Yeah, exactly, Alejandro. Positive, because the negative charge inside the membrane is going to attract this positive ion that's going to allow it to go forward, or that's going to increase the likelihood of that ion wanting to come through. So it's not a hard rule just because there is a negative and a negative here. That doesn't mean that the negative can't go through, because remember, most of the time we are dealing with like only ions. So you have to play. It's like another factor. So you have concentration gradients and electrical gradients. So if you have like a super high like negative ion concentration here and like super low here, that can overpower the amount of charge, the difference in charge. So the negative ions can't flow through if the concentration is high enough. But if you have like, let's say, equal amounts, then obviously the one with the opposite charge is going to be more favorable. Does that make sense? It's just like another variable you have to think of. So in addition to, okay, is there like what's the different concentrations? Well, let's think of the charge. Like which one is more likely to go through? She doesn't focus on that too much, but for stuff like in the future classes like biochem, you have to like use math and like calculate the energy or like which one's more likely, stuff like that. But you don't have to know that too much for your exam, to my knowledge. But let me know if you guys need any more explanation, but I think you should be good. I think that's good.
We're back. It's David, back at it again. So Joanna Gardner asked, what is the role of second messengers in the signal transduction pathway with reference to the phosphorylation cascade? That's a great question. So just to implement you guys, let's go inside a cell. So right here, we have our adenyl cyclase, which converts what to what? Tell me. Adenyl cyclase converts what into what? What into what? Yes. Wait, Meredith. No, Meredith. It's 18. They said 18.
Yeah, that's right.
You got it, Meredith. ATP, cyclic AMP. And from there, cyclic AMP will be used as the second messenger. So going back to the question, the role of second messengers in the signal transduction pathway in reference to the phosphorylation cascade. The CAMP is the second messenger of the pathway. So following this part of the pathway, we have our phosphorylation cascade. But in order to initiate the phosphorylation cascade, this first protein kinase needs to be activated. So in order for this first protein kinase to be activated, this CAMP, which is our second messenger, will bind onto, yes. Will bind to our first protein kinase, which then activates it. So to clarify this, in terms of the whole pathway, so let's say we have our G protein coupled receptor. This is going to be our ligand. It's going to be our ligand. And then it's going to be a G protein coupled receptor, and then we have our G protein. In this case, the first messenger in this whole pathway would be the ligand, the first messenger. The second messenger it's carrying out the same signal and function as the It's basically an extension of the first messenger. But if you guys remember, what's the point of this in terms of let's think of this in the broad scheme. So we have one ligand. And what is the purpose of the signal transduction pathway in terms of what it does to the signal?
Signal, I'm assuming.
Yeah.
Signal. Yeah.
Yes, Jessica. Yes. Signal. Signal amplification. So how that happens is with going back to the second messenger, which is an extension of the first messenger, aka the ligand. The second messenger is made in mass quantities. These will bind to different protein kinases, which will then activate them. The activation of those protein kinases will then fall down through the protein kinase cascade, which is, the role of the protein kinase, which is to phosphorylate. So this protein kinase will phosphorylate this protein kinase. And what's phosphorylation? That's right. Phosphorylation is adding phosphates to something. So this protein kinase is going to add a phosphate to this protein kinase, which then will activate it and add another phosphate to this protein kinase. So as we said, this is only one path. This is only one cascade. We have many cascades going on simultaneously, which then turns our one signal into a huge many signals. Okay. I hope that answered the question. Now, someone asked, can you go over action?
Oh, wait. I have a question. So we know that these protein kinases activate tons of other molecules, right? And we know it's not just one. It's not a linear progression. So this protein kinase can activate tons and tons more of other kinases to amplify a signal, correct? But so we amplify it. So what? What is the result, or what is our response going to be after all the amplification? Like, most people just like kind of focus on the signal and the secondary messengers and the amplification, but what happens at the end of that? Let me know. Let me check. Just to clarify. So yes, it is. You are creating a fast response because of your secondary messengers, but what happens after you've created a ton of secondary messengers? So basically, what do all those protein kinases do? What is the ultimate goal of cell signaling usually? Does that make sense? Okay, yes, Alejandra, transcription factors, exactly. So at the end of the cascade, you'll typically activate a transcription factor, which we know will then go to the nucleus, help bind to the promoter, or bind the RNA polymerase to the promoter, and then you can control your genes. So remember, to typical gene regulation, you're always controlling transcription. So most of the time, you can think of it as if you do transcription, you're going to express that protein. So if we have a bunch of these signal molecules telling the promoter, telling RNA polymerase to bind to the promoter with transcription factors, basically you're doing the signal to help create transcription factors to increase the likelihood that polymerase 2 will bind to the promoter. The ultimate end of this is going to make a protein. So you signal to turn on a transcription factor to make a protein to do whatever you need to. Good job, guys.
Okay. And so Karen Yun is asking, does it go to the nucleus? Yes, the transcription factor 10, yes, it does. It goes inside the nucleus to bind into the DNA, and the DNA is located in the nucleus. And now, so Njeri asked, can we go over action potentials? We can. It's a big topic. So instead of reviewing the whole thing, I'm going to do something very condensed. And what you guys will do is ask me specific questions so we can clear up the mishaps.
Also, in another video, I believe it's cell signaling. David Hernandez also went over action potential. So if we don't have time to, or we don't answer your questions properly in this, go check out that other video, and I'm sure you answered it in there. Emmanuel. Okay, so while David is drawing, Emmanuel, so for the histone proteins, there's like, wait, H2, 2, 3, 4. Yeah. There's a really good picture in your book that shows you which ones, like, you don't really have to know much about them. Just like, I would be familiar with the numbers like H2, H3, H4. I may have missed one. I know there's also H1, but definitely know like what it's in the octamer. So there's octamers, so 8 total proteins, and there's 2 of each kind. So I would know those. And then there's also 1 histone protein that is not within a bundle. And does anybody know what that is? Do you have headphones? Oh, you do have headphones. Can you grab me? Okay, so super ghetto demo here, but you have 4 proteins. 4 proteins total, but there's 2 of them each. I don't have it here because we don't have enough markers, but there's 2 of them each to form an octamer. So think about basically just double each color. The black, the little white eraser tip, brown, and orange. And that is within your octamer. And octamer means 8, 4 times 2, 8. And then also you have another protein, H1, which is not it's not within the octamer, but so you have this, and then you see my headphones wrapped around. That's the DNA wrapped around my histone protein, and you also have H1, which is my chapstick here, just associated with it nearby. It's not within the octamer, but I believe it's probably actually like, that's a terrible demo, but it's just nearby, and it helps like keep the DNA or help stabilize the rest of the structure. It's just, you just have to know like it's not within this whole bundle right here. I mean, I hope that makes sense. And then now David's back with action potential.
I'm back. So what I have here is what I have drawn here. I apologize. Is a graph of the change in membrane potential throughout the action potential. So I have here 5 different stages. Can you guys tell me what these stages are?
You back on?
Hello? So Kevin, you've almost hit it right on the money. Injury, yes, that is A. A is resting. Almost right on the money. Okay. This is actually, I apologize, this was a bit rushed, so I didn't actually draw it as well as I could have. What I meant to do was split this one into 2. Let's see.
You have to redraw that.
So A is resting. B is threshold. C is depolarization. D is repolarization. E is hyperpolarization. F is returning to resting. So, a quick thing because we're actually running out of time, but just to get through this about resting membrane potential, what we have is our electrochemical gradient. Our electrochemical gradient, which is maintained by F is returning. Meredith F is returning to resting membrane potential. And we'll get to the details of that. But at resting membrane potential, we have our membrane potential, which is maintained by our sodium-potassium pump, which helps to keep the gradients of sodium and potassium in their respective sides. So what happens at threshold is that we're going to have some type of signal. So like, let's say for our eye, in order for the action potential to occur, we need a certain influx of sodium to come in. So in this case, with our normal, regular, ungated sodium channels, we need a signal strong enough in order to be activated, in order for the voltage-gated sodium channels to be activated. So in the case of your eye, like let's say if you're squinting, you're not really getting that much stimulation or that much light into your eye. As you open your eye a little bit more, you're going to get more light's going to hit your eye, and specifically on your photoreceptors. And once you fully open your eye, you're going to receive enough light on your eye such that sodium channels are going to, enough sodium is going to influx into the ungated channels such that the membrane reaches the proper membrane potential for the voltage-gated sodium channels to open up. Once the voltage-gated sodium channels open up, we have a high concentration of sodium outside the cell. So that high concentration of sodium outside the cell will start rushing in because we have more sodium channels opened up. More sodium channels opened up means more permeability for sodium. As the permeability of sodium increases, more sodium comes in, the membrane potential increases. It increases until it hits right here, the action potential, which then the sodium channels close. When the sodium channels close, which channels open up? Can you tell me, Derek?
I believe it's potassium.
That's correct. The potassium channels open up. So as the potassium channels open up, let's see. Let's go back to our diagram. So we have both sodium and potassium are both positive charges, which is kind of freaky. And they're moving across the membrane. So how could, if you think the movement of positive charges would equate to an ever rising membrane potential, but that's incorrect. It depends on where those positive charges are. So in this case, we have sodium flying in before our action potential. Sodium went into the cell. As positive charges enter the cell, the membrane potential increases. Now our potassium ions, which are also positive, are leaving the cell. So as potassium leaves the cell, it becomes less negative. It becomes less positive. Less positive means more negative, which is why the membrane potential drops. But as you guys remember, there's a big difference between sodium channels and potassium channels. And what is that? Voltage-gated sodium and voltage-gated potassium. And what's that main difference? That's correct. Sodium channels have what we call an activation loop. Voltage-gated sodium channels have an activation loop. That inactivation loop. That inactivation loop allows the sodium channels to close very quickly. Hence why when we hit this membrane potential, it closes immediately. Potassium channels, on the other hand, don't have that activation loop. So once it reaches the resting membrane potential, it starts to close, but not completely. And because of that, they get a bit leaky, and we have more potassium leave the cell than anticipated, which is why it hyperpolarizes. Hyperpolarization. Step B. So as we hit hyperpolarization, we need somehow to get back to resting membrane potential. What protein do we know is possible? What protein's function do we know is to restore the gradient of the respective ions within the cell? That's correct. The sodium-potassium pump. So right here, our sodium-potassium pump, going back to Meredith, what you were saying with at F, returning to resting membrane potential. Since we're hyperpolarized, we need to return sodium and potassium back to their respective sides. Because right now, we have too much potassium that left the cell. So in order to do that, we get a sodium-potassium pump, which pushes potassium against its concentration gradient using what? ATP, that's right. We're using energy to push things against their concentration gradient back to the respective places. And once we push everything back to the respective place, they return to resting membrane potential.
Kevin got you.
Kevin's coming through.
Yeah, so this is kind of an abbreviated version of this. And like I said, David Hernandez did one on the cell signaling video. It's very in-depth. And he spent a lot of time on it with live drawings and not just words. But if you're confused about what David said about positive charges leaving but the membrane potential getting negative, hey David, if I give you $3 and you give me back $2, what's my bank account?
If you give me $3,
yeah,
and I give you back $2, does that mean you're -1?
Yeah, it means I'm -1. Does that make sense? That's kind of how that works. Yeah, so I don't worry about the numbers too much, but basically you give out more than you lose. All right. It looks like we are out of time, but thank you guys so much for tuning in. I hope you enjoyed. Give us some feedback. We'd love to hear what you have to say. Tell Dr. Latham David will be in class for your exam one day. But good luck. Good luck on your exam. Signing off.
What is it?
All right. Yeah.